I joke with people that if you had told me when I discussed with Nubar in the early days that I would have to raise $5 billion, I have 100% chance would have not have taken the job because I would have been petrified. There's not a lot of sugar coating in my family. And so I'm a bit the same. I'm an engineer, so I like facts. If I believe it's black, I'm not going to tell you it's gray or white. And we have around 10 molecules for other rare disease in the liver that really become copy and paste like we've done with all vaccines. Doing the first one is like building a new industry, scientifically almost. But then doing the second one is almost like cheating because you just change the sequence, which is just information of the gene. The team has already written 2,000 GPTs within the company, and they are literally across everything. So we need another podcast one day.
I will get back to you. Bienvenue to a new episode. I'm your host Philip, and on this show, I'm interviewing the best Europeans in biotech to help you grow. Today, I'm in Boston, more precisely in Cambridge and Kennedy Square. I will talk with Stéphane from Moderna. That's the headquarters. Stéphane co-founded Moderna in 2010, raised $5 billion, helped solve the pandemic, and now the company has 3 products approved on the market. I've known him since 2014, and he is one of my biotech heroes. We will talk about the lessons he learned from this extraordinary journey. We will also talk about what's next for the mRNA field and how AI is impacting so many parts of the company. So, here's my conversation with Stéphane, and please hit the like or follow button if you're enjoying it. Welcome to the show, Stéphane.
Thank you so much for having me, Philip. It's good to see you.
Yeah, it's been a while.
Yes.
So I want to start with personal lessons. And actually, one thing I quote you a lot, and also on the show, is you told me that biotech is about people, as real estate is about location. So can you expand a bit on this?
Sure. I think it goes back to observations I've had building the company, which, as you know, there was nothing initially. And then the team worked on the science. And then when we had good science, we worked on manufacturing process. And then we took the drugs to a clinic, 1 drug, 2 drugs, 4 drugs. And then the pandemic happened and we rallied the troops. And if you look about it, all those things have been done by people, obviously. And one of the things I've realized is that the culture you set, the culture fit between the enterprise and the people you hire. And I think it's really important as you hire people to make sure they fit your culture. Every culture is different. And there's not good or bad culture, it's just a culture. It's like families. I think families have different cultures. But I think what's important is the fit. And so I think you need to make sure as a leader to really set up the culture, to reinforce the culture all the time, to talk about the culture so that you get the best people be able to work for patients.
Yeah. And I remember one thing you told me as well was like you preferred A team and B science than A science with B team.
Yes.
Because the B team can screw up the A side.
Yes, that's correct. And actually, I
studied Bob Langer.
Yes, exactly. I was going to say I studied from Bob Langer. In the early days of Moderna, I remember Bob being invited to meet some investors in Moderna. That's for the Series C, so we're talking literally spring of 2012, really early.
Yeah.
And one of the things Bob told the investors, I forgot the question I was asked, but I remember his answer precisely. He said, early in my careers, because of course he's a scientist, Bob Langer, he's like, I used to believe that the most important was to have A science. And what I've learned over the years, as he built 40 plus companies, is that you better have a B team with A science versus having a B science with an A team.
Yeah. Yeah, like that. Switching a bit, also one thing you told me, I think, was you talked about the Boston Biotech Mafia, a bit like the PayPal Mafia, I guess, in Silicon Valley. Can you expand a bit on this? Like,
Sure. So for people listening who have never been to Boston, you had the chance, Philip, to be here many times. It's actually Cambridge. It's actually the Cambridge Mafia. Because we're literally on Cambridge, not on Boston side of the river. And MIT, you can see from your seat, is 2 blocks away. Because Moderna now is big, it's only us in this building, but we actually used to be in the building behind you.
And this one, no.
As well, we had 5 or 6 buildings as we keep growing, but we started here, and this building is full of small companies.
Yeah.
And so by definition, you bump into people, you go to restaurants, you go to meetings, you see people. And then people leave companies and they start another one. And so if you have been in this ecosystem for long enough, there's a lot of connection, there's a lot of friendships. That's called the Biotech Mafia of Cambridge. I say Boston from when we are very far away from Boston because most people don't know the difference. And that's sort of a uniqueness here is that it's easy. I've spoke to a lot of colleagues in California, especially in the tech world, and they say Silicon Valley is not the same because you have to get in the car. Well, literally here, you can literally go down the building and just go to get a coffee, at Starbucks or whatever. You're gonna make you might meet 5 other CEOs and 2 professors from MIT.
That's funny. I mean, yeah, what's amazing is that there's a density. Kenneth Square is whatever 3, 4 miles, 5 miles. And they call it the highest density of brains.
And it's true if you look at all the molecules that have come. I mean, Alnylam, is 2 blocks away this way. There's Biogen 1 block away. There's the AstraZeneca building. This is Amgen building. And then all those startups, it's incredible. And literally 2 blocks away, as we said, it's a Koch Institute where Bob Langer and all his team occupies the top floor. And you have so much innovation there. So I think the academic world as well has incredible innovation, which a lot of time becomes companies.
That translates it. So you're born in France or French in the US, and I heard from a common connection that sometimes you're more American than French.
Yeah.
How do you feel?
Yeah, I would kind of agree. So it's interesting. I've always loved business. I come from a family, we talk about culture initially, about business culture, but I come from a family where we say things as we believe them or there's a lot of, sugar coating in my family. And so I'm a bit the same, which is I say things. I'm an engineer by training, so we're all biased by our own trade. And I'm an engineer, so I like facts. And so if I believe it's black, I'm not going to tell you it's gray or white. I'm going to say it's black. And if you say it's gray, say no, it's black. And so it's true that when you look at American business, there's a side of it that is really similar to that world, where you look at facts, you make decisions, you pivot quickly. But I think it's something also required in science. I think one of the success of the culture of Moderna is the ability to pivot the strategy of a company quickly on new data, and not to be scared to say we were wrong, because before we believed something else, data-wise and science-wise. And with these new findings, the previous strategy is not the best strategy, and it will change even if you look like a fool because you change strategy, or what you used to say was right is actually now wrong, or vice versa. But that's the piece where I think the focus and the obsession on the mission to help patients, if this is really what you care about, looking like a fool a few times a week or a few times a month is fine, as long as we help patients.
Yeah, that's the most important. And I'm curious you've never wanted to come back to Europe. I guess you travel a lot there, but like coming back or relocating to Europe,
people ask me the question regularly. I don't think it's a practical question, so I cannot give a practical answer, which is, we have 5,000 people here in the Massachusetts space. The company was started here because, as you recall, it was a set of founders from Harvard and Bob Langer from MIT and Noubar from Flagship. So you have 3 academic co-founder and 1 kind of entrepreneur co-founder with Noubar. And so everything is here. Most of the board is here. So I just don't see a practicality where I can go to London or Paris or Berlin. It just cannot happen.
maybe to retire.
But that's a very different question. And that's not for today.
Not for today. Curious on also I think I remember you told me I want to build the next Amgen or the next big biotech. How is it still how is it still true?
So if you recall, because we have a benefit, unlike some of the listeners, to have known each other for many, many years, since the first month of Moderna, I told all our stakeholders, as we were hiring employees, hiring board members, raising money, I told everybody, I said, look, because mRNA is information, we're trying to build a platform, but at the end of the day, we're going to have no drug or a lot of drug. And I used to tell everybody a long time ago, it makes no scientific sense it would be a 1 or 2 drug company. It would be 0 or a lot. So the good news now, as we tape this discussion, we just got another positive news from FDA overnight. If you look with the approval of a next-gen COVID vaccine mRNA 33 called mNEXSpike, which was approved by the FDA at the end of May, we have now 3 BLAs for different mRNAs. They are all very different mRNAs, different chemical entities, different sequence. And so we are already 3 molecules. We said we should have a flu data phase 3 efficacy in the summer. We've already shown the flu COVID combo antibody data, which looks amazing last year. We're waiting for CMV, waiting for neuro. We showed, 3-year survival data in cancer that I hope we can talk about that are so exciting to me. I think the next frontier of Moderna is in oncology. And so it's not going to be zero drug. So we know it's going to be a lot. So is life a straight line? No. Of course, during COVID, we had to, like our partners at Biotech, our colleagues at Biotech, to do incredible things to get the vaccines out so fast. But now, if you want to end the time of kind of really focusing the company on the key programs, the priorities, building, rebuilding, launching, managing the sales decrease post-pandemic, quote-unquote hangover, because we always knew that, I used to tell the team as we are building the manufacturing capacity, I say one day we're going to have to climb down the mountain that we're building right now. Because it was obvious that you don't need everybody to get a vaccine every year. Actually, in 2021, I got 3 shots because I got the first 2 plus a booster. And I always say people, it makes no sense that I will get 3 shots every year forever. Yeah. So you just do the math plus people who don't want them. So we came down the mountain. We are reclimbing the mountain now with 3 products approved and the cancer data and all everything. So I think the next few years are going to be really exciting. Because I think the platform is really coming to music in the next few years. Because you could see Moderna in 26, 27, having 6, 7, 8 products approved, including infectious disease, oncology, but also rare disease. We're now in phase 3 in oncology, in rare disease, in pivotal studies. And so 2 years is going to come pretty quick. Yeah. And in 2 years' time, we could be again 7, 8 products approved.
Yeah, that's amazing. We'll get back to this.
Sure.
I want to talk a bit on a bit personal, personal question. You don't need to answer it, but on your shares, you own quite a lot of shares of Moderna, and obviously the stock price went up crazy. And then, I guess your virtual net worth as well. Like, how did it feel? Like, you also, kind of climbed the mountain, climbed down as well a bit. Like, how did you, first feel about it?
Sure. So the good news is I've never been driven by money. So for me, money is a consequence of success. It's not the goal. My wife and I have said publicly many times that we're going to give most of our money away, which we started doing because during the pandemic, we had actually one of my first big stock option grants on Moderna. I wrote a blog about it. That was going to expire, so I had to sell the shares. I had to exercise the option. And so then we sold the shares and we gave 100% of the proceeds after paying our taxes to charity. Okay. And so the yo-yo of the stock price, I think, only matters to people if you care about money a lot and if you care about selling, because like Warren Buffett says, the stock price is irrelevant if you're not buying or selling. Yeah. And given I have a very long-term view on this company, given, as we just spoke about, we already have 3 products approved, I think we're having 7, 8 and more, and I'm not a seller, is that actually I bought stock in February. I bought quite a lot of stock with the cash I have personally, because the price at which Moderna is in those days makes no sense. And as you know, with stock market, there's always fears and greed. And either you are overvalued like we were during the pandemic, or you are what I believe is undervalued right now. And so it's just years of life. I always remind the team, because sometimes it's disturbing to employees, because everybody as a company has stock option. Yeah. I made that a policy since day one. The secretaries, the people working in the factory, on the shop floor, everybody has equity at Moderna, because I want everybody to behave like an owner and to share into the success of their work, which, as you say, is very American. And sometimes I talk about it with people who say, look, when the stock price was very high during the pandemic, it made no sense.
It was like higher than whatever GSK was. I mean, he went off 100 billion market cap.
Yes, and sometimes it was crazy because I remember there was a month, I think it was in summer of 2022, where the stock price doubled in a month on no news. Like there was literally no news, no press release by the company, no new scientific publication, like zero news, and the stock price doubled in a month or a month and a half. And I kept telling people, this makes no sense. Don't get too excited. Put your head down, do the work, do the science. And I tell them the same thing now that the stock is not in a good place. Yeah, it's low, which is don't sell your shares now. Do the work, do the science. The stock price will catch up. Nobody should be driven, in my opinion, by the stock price. We should do the right thing for patients. If we do that, as a consequence, we'll make profit, and that will create shareholder value for stock price.
Yeah, that's good. Let's see this. Maybe just a quick question on a bit on your career, because I think you started more when you studied in France. You started in France. You worked at Eli Lilly, I think a bit in the US as well. You went back to France, bioMérieux, you're a CEO. Like, because like, what did you like? How did basically, if you look back at it, how did all these roles lead to where you are, to Moderna and where you are now? Like,
yeah, it's a great question. So the piece I've been lucky is since I was maybe 16, 17 years old, high school, I always dreamed of being a CEO of a technology company one day. I don't think I have a big ego, so I would have been happy if by the end of my career I would have run a company of 10 people. Literally, I've been it. But the piece I wanted to do one day, and I think because it's how my brain works, I'm an engineer and I'm a system thinker. And I always dreamed of the idea of, by being the CEO, having to make sure all the parts make sense together. Because if you have a good science strategy, but your manufacturing strategy is not aligned with it, or either your commercial and/or your financing strategy, you're going to fail because all those things are linked. And I like the fact that I had this belief that the company is basically this system where the CEO has always to kind of rebalance, because either be outside environment or the scientific data or the customer performance on sales will impact the system, and you have to rebalance the system in real time over time. And so that was kind of the thing I think I wanted to do. And so I've always asked myself the questions for my career, say, how do you get there? And so I started after my engineering degree, which I did a bit in France and a bit in the US, in Minnesota. I went to Japan at bioMérieux, because for an engineer, it was I wanted to be in life science. I always thought that life science was this great industry where you use your time to make products to help other people. And I just think it's like super cool to be able to use your time to do good things in the world. And so as an engineer, it was kind of an easier route to get into life science. And so I learned a lot about diagnostics. And then I got my MBA to the US. Then I joined Eli Lilly because I wanted to go into the treatment side of the continuum of diagnostic and then treatment. I had a great run at Lilly. It is a great company. Very great company. I worked for him in the UK, in Indianapolis, US, in Belgium. And then Eli Lilly, the chairman of bioMérieux, called me and told me that they were looking for a new CEO. And so they ended up giving me the job. And so I was...
You were quite young.
I was quite young. I think around 33 years old-ish, if I remember. And so I went back to diagnostics, and then the founding team of Moderna was looking for a CEO. We had one employee, and so they wanted to put money in and start building a business. And I've known Noubar Feyan, who is the chairman and was the co-founder, or is a co-founder of a company, who when they talked to me about using mRNA as a drug, he knew I'm a biochemical engineer by training. So my first reaction to him is like, you're crazy, you're going to inject mRNA in people and they're going to make their own protein like a bioreactor. He's like, yes. I'm like, you're crazy. And he's like, I don't know, maybe.
I heard you had this discussion on the bridge.
Yes.
Look for the bridges.
Exactly. Okay. And so all those pieces led me to Moderna because I learned the CEO job at bioMérieux. I always tell people, I don't know if I would have been successful in this job as CEO of Moderna if I had not been CEO before. Because it's so hard. I tell people it was harder to do the CEO of Moderna job than the CEO of bioMérieux job. And the CEO of bioMérieux job was hard because it was in a large company in 40 countries, 6,000 people, publicly traded, a lot of product line. It was not an easy job. But this job was 10 or 100x harder because the science, there are so many unknown because nobody had ever done mRNA drugs before us. Raising money, I'd never raised money before. And we raised $5 billion in the Moderna history. And I joke with people that if you had told me when I discussed with Nubar in the early days that I would have to raise $5 billion, I've 100% chance would have not have taken the job because I would have been petrified. I've never raised a penny in my life. I don't know if before us, actually, I think no companies before us will raise $5 billion. So you have told me you have to raise $5 billion and nobody has ever done it. And I never done it myself. So I think I would have just freaked out and told Nubar, you're way too crazy, find somebody else, I'm not doing this thing. So I think sometimes for entrepreneurs, I think not knowing is really important. I heard recently a podcast with Jensen, the CEO of NVIDIA, and he was asked a similar question. And he said, yeah, I will not do it again. With what I know now and all the pain it has taken us and me to build this thing over the last 30 years is their journey. Because I started in 93. He's like, I will not do it again. And he's like, and he made a comment which I agree 100%, which is there's a naivety of entrepreneurs because most of entrepreneurs have never done it before. Some are several entrepreneurs who have done it before. So maybe they're superhumans or they are masochists or they are crazy or I don't know.
I was about to say.
Yeah. But if I had not been CEO of bioMérieux, there's no chance I would have done that. And if I had not worked at Lilly, and there's no chance I could have done this job well. We might have been bankrupt.
And looking back, you would do it again, Moderna? As Jensen said.
I will do it again for only one reason, which is, and it's the same reason that I did it the first time, or the real time, which is, it was a crazy, crazy decision. All my friends and family told me not to do it, except my wife, to be fair to my wife. And the way I thought about it at the end, after spending a few weeks talking to Nubar and the founders, talking to a few scientists to get a bit of a sense, because I'm an engineer, I'm a biochemical engineer. I understand a bit the space, but I'm not a PhD in molecular biology. And so I had to talk to a few experts and to help make my decision. And it was clear that the odds were really against us. Because already, as you know, starting a company in any field is hard. Most companies fail. In biotech, it's even harder because it takes so long and you need so much money. And doing a biotech new company with a new technology was like on the borderline of insanity. And I told my wife, she's like, look, it has maybe 5% chance of working. And so the reason I did the first time is I did because I believe that despite the very, very low odds of being successful, if we're being successful to the point we started our discussion today, it was going to be a platforming. We're going to make a lot of drugs. And the mental model I had in my head, which has served me a lot to play the long game and to try to be thoughtful about the strategy, is to say, if you think about it, recombinant technology, the biotech industry, I started in the 70s with Amgen and Genentech and then Biogen and a few companies. But if you look at what it has done in 50 years, because now it's 50 years after, and what it has done amazing in terms of new protein-based vaccine and the insulin and the Keytruda and the checkpoint and then I could go for hours, that now 7 out of the top 10 drugs sold by pharmaceutical companies are biotechnology product drugs. The impact on humanity has been gigantic, and it was clear to me because of the biology, because we are at this amazing, exciting time where we learn more and more about human genes and what they do and how the human body works and so on, that over time we should be able to find ways to get mRNA into a new cell type. And once you're in a cell, then it's easy, just molecular biology. It's very logical. It's very deterministic. And so it was clear to me, again, what I told you, which is it was going to be zero or a lot. And so the opportunity to go and try to work really hard with a lot of super smart, dedicated people to do something that could impact humanity for 50, 100 years. It's like, I'll sign again today. Even now I know the pain and that it will not be fun every day. But the journey is so worth it. And the personal feeling of helping other people is such a amazing, unique feeling. Because the mental model I always had, and I tell my team all the time, is I think about the people I love in my life, my wife, my kids, my parents, my friends, my best friends, and so on. And I use that mental model as to use for people you love. There's a lot of people in your life that I've never met. I most probably never met them in my life, but that you care a lot about. And I always believed, for the way I was raised, that I don't love the people that I love in my life more than you love the people you love in your life. And so I always use the mental model of people I care about deeply, as I want those people to be protected with vaccines. I want people to have treatment when they have cancer. We have a family that has a lot of cancer history. And so I've always used that mental model to think about how do we really push hard. And I know sometimes there's been a criticism of our culture, but it is what it is. How we really push hard, because every day matters, because people are going to be waiting either today or in 5 years' time. And if we're late by a day or a week, those people, which people in their life love them, won't have a product that we should bring to them.
Yeah. Actually, it's funny you mentioned the culture. I had one thing about this year. I like what you said about culture is different. It's what's important is the culture fit.
Yes.
As you said, the culture is a bit hard. I think you mentioned intense.
It's demanding, yeah. It's demanding for sure.
It still is, yeah.
Yeah. For the same reason that it was on day one, which is the mission is what drives many, if not most of us at the company. That I really believe deeply in my guts, in my brain, in my heart, that every day matters. I don't want, again, I use my family as a mental model for everybody. I don't want my wife not to have a cancer drug we're working on if she has multiple myeloma in 5 years because we are a year late, because we didn't do our best work. If we do our best work, I'd be able to look at myself in the mirror in the morning when I shave. Say, yeah, I really believe with your best work, we took the right risk. We push each other to get the best idea to win, not who was the most senior person or who was here the longest or whatever. Those type of things I don't care at all about. And so that demanding culture is demanding on ourselves and of each other. And I would say people is be demanding of me. I believe I'm super demanding of myself, but be demanding of me, push me, challenge me. And I wanted this culture where the best idea wins. And I always said, new employees, I don't care if you've been in the building a year or 10 years, if you're a technician or vice president, speak up, debate the ideas, because we are data-driven, fact-driven, best idea win culture. And you need to speak up, because if you don't, again, somebody, maybe in 5 years or 10 years won't have a drug they need because you didn't speak up.
I like that. So, yeah. Transitioning a bit more to the, let's say, mRNA industry. And one thing, you just mentioned about the science that when you started, it was crazy, mRNA, that it would work.
Yes.
And I think when you started, there's not too much, but I think Ingmar was on the shows of the founder of CureVac.
Yeah.
I think he had started a few things.
A few years before, I don't remember exactly when actually, no. You know what? They started a lot before. We started in 2011.
Yeah.
So a decade before we started CureVac.
But he started really from zero.
From scratch. We started from zero too. People forget, but the first round of Moderna was a $2 million round.
Yeah.
So, much later in the company's life, when we were in the clinic, we did very big rounds that we were known for, kind of a $500 million round every 12, 18 months. But people forget the first round was $2 million.
And the first coffee machine was called Pascal Norsaurio. Yeah. I'm curious now on the science, like, if you look back, I think let's say on the paper, and when you describe it, you inject the mRNA, it produces a protein, like, it's logical. If it works, it's amazing. Can you say a bit more on what were the biggest, like, science hurdles?
Sure.
That were overcome?
Yeah, and there are a few that were known by everybody almost in the field that I raised to Nubar during that very first discussion about using mRNA as a drug. mRNA is known to be highly immunogenic. Our immune system has developed tools over thousands of years to protect ourselves against viruses, because as we all know, there's a lot of mRNA virus.
Yeah.
And so usually when your body sees a foreign piece of mRNA, you think it's a virus and it gets very upset with it. That's how we survived as a species. So this I was, of course, aware. So I told him about, mRNA is highly immunogenic. He's like, I know. And then I told him, mRNA is very unstable. He's like, I know. I said, okay. And so there was a lot of technical hurdles, which is how do you stabilize the mRNA? How do you reduce the immunogenicity? So the reaction of your immune system. And then how do you get it to the right cells? I think some of the work that had been done in the labs by Rossi and Carico and Weissman and others over the years was in Petri dish. So you don't have a delivery system. You don't have an immune system in a Petri dish. You see what I mean? You have all the same cells from the same tissue. So it's a very easy system, but it's a place to start, obviously.
Yeah.
You need to start simple before you go complex, obviously, in science. And so we had to take this from a very simple concept to the hurdle of a very complex human immune system. And then to get mRNA in the right side. To give you a sense, one day I remember, it must have been 2014-ish, or must be 3-4 years old, or maybe it's 15, I forgot, where we had a massive issue in our rare disease program in animals. It looked amazing in rats. Amazing. So it was injecting IV into a rat, an mRNA, to go to the liver, to replace the protein that in the future a kid, now a rat, was missing, so that the rat liver will be functioning normally like your liver and my liver. Okay, yeah. So we're correcting a protein deficiency. It looked amazing in rats. So we go to monkeys. It doesn't work. And we don't know why, and we repeat it, it doesn't work. And then we do a lot of histology work. We look at, tissues and so on. And we realize in the liver, the mRNA was coming through the central vein, of the liver. And then you will go into the first cells that go after the vein, which is the Kupffer cell, before getting into the next layer of cells called the hepatocyte. We didn't need to come to the hepatocyte. In the monkey, it was stuck in the Kupffer cell. So you go, you will go IV into the vein of the body through the blood. You will go into the Kupffer cell and it was stuck in the monkey, which didn't happen in a rat or in a mouse. So it took us a long time to first figure out what was the problem. And then it took us even longer to figure out how to fix it. Because remember, you model to start optimizing the technology is again the mouse and the rat, which you already learn behaves differently than the monkey. So it's type of things that makes the difference between a cool scientific idea in people's brain or in a petri dish in the same cell system to take into a living system and to scaling it across species size, which also raises the question. Now, the good news, we are sitting in 2025 and we know it works great in humans, which is why we're in phase 3, because the phase 1, 2, the kids, some of them have been several years on drugs, have a 70-80% reduction of hospitalization because of a drug. So it's working. And when you hear a patient story from the parents, that are quite amazing and touching stories. So that's why now we're in the phase 3. But if you look at the journey of that technology, it was so hard. The good news now, because again, mRNA is a platform, mRNA is information. We have a second one behind it, and we have around 10 molecules for other rare disease in the liver that really become copy and paste like we've done with our vaccines. And that's where you're going to see the power of the technology. Doing the first one is like building a new industry, scientifically almost. But then doing the second one feels almost like cheating, because you just change the sequence, which is just information of a gene that's available on the internet, and you have a new drug and you write it again.
Can you just go a bit deeper into how did you reduce immunogenicity? How did you improve the stabilization? Yeah, how did you improve the delivery?
So there were a few big scientific steps, and there were dozens or hundreds of small ones. So I think it's tough to really tear all those things apart. One that is important and well known is the modification of a uridine that was initially discovered by Carico and Weissman, for which we took a patent from UPenn years and years before the pandemic, which, as you know, they got the Nobel Prize for their work, which was clearly an important stepping stone of the whole puzzle of how do you make mRNA work safely in human and get into the right cells. There was a lot of manufacturing know-how that we learned by doing it, because you make the mRNA and then you inject it and it's highly immunogenic, and you study what is in a sample of your manufacturing product, and you realize there's a byproduct of an enzyme that create little pieces of mRNA that actually excite the immune system.
Okay.
And so you can separate them by doing purification, and you can test them together or separated, and you realize that when you take them out, you have less immune response, and you keep at it. So there's a lot of manufacturing know-how, and theoretically we use machine learning back in 2017, so pre-COVID and pre-GPTs and so on. We use machine learning to evolve some enzyme to remove some of the issue we observe that exists in nature. So we invented enzymes that don't exist in nature for manufacturing in our plant. That was another part of technology.
And I guess it was important to have your own manufacturing sites.
Correct. There's a lot of technology around, optimization of the mRNA, because as you know, there's a lot. The tail, the URL, 3', 5', the cap, which kind of the nose of the mRNA. So there's a lot of piece on the mRNA molecule itself. Then the formulation, which both protects the mRNA from degradation in the blood, because mRNA just injected directly into blood is degraded in seconds. Because again, through evolution, we have enzyme in our blood that will just chew up mRNA like candies very quickly, so we don't have time to replicate and become dangerous. And so part of a delivery system that we developed, one of the goals was to protect the mRNA by putting the mRNA inside like a protecting layer, if that makes sense. The other part was to carry
the LNP, or
sorry,
what? That's LNP.
Yeah, for example, that's a lipid nanoparticle, the LNP. The other function of a lipid is to take it to the cell where you want the mRNA to have its biological action by making the protein you want in the right cells. So there's a lot of, so that's why there was, again, it would be unfair to scientists, but there were some big stepping stones and dozens, again, maybe hundreds of small stepping stones. And by the way, we're not done.
Yeah.
It's only 20-year-old technology. We're still investing $200, $400 million per year on basic science to keep pushing the boundaries, get mRNA into new organs, get into new therapeutic area like autoimmune disease, for which we have nothing in the clinic yet, but we're still working in the lab. So we're not done. We're still getting started.
That's amazing. I'm curious on the LNP and the delivery, because I think that's at least what I hear is one of the major challenge in whatever gene therapy or cell therapy delivery. And what you said, you want the mRNA to go to the right cell. What I heard with LNP is that it still goes mostly to the liver. Is that true, or can you really modify
So
that?
it depends how you build them chemically. It depends what you add around them that can bind to something in your body, like a receptor. Like you can put, antibodies or ligand on the surface of the lipid. And it depends how you inject it. Like for example, an example that listeners to a podcast will know is when you inject mRNA as a vaccine in the muscle of your arm. What we learned years ago in monkeys first and then confirmed in humans is that when you inject it in the arm, a small percent of the mass of mRNA makes the protein in your muscle cells, like around the site where you have your needle injecting it, as you would expect. But most of it drains to your lymph node, into your armpit, into your immune cells, and actually gets into your immune cells. Very little goes to the liver. If you inject it like this, if you inject it IV with a certain chemical structure of your lipid and a certain size, because the size also is another key process parameter, because depending on size, it goes to different places because it has to get somewhere. It has to get into something, if that makes sense, for the non-technology person. And so those are all tricks that we learned. So we studied cell shape, size, pores, chemicals, proteins on cell surface. So it's just a lot of pieces. Which is why, again, go back to your previous question. I think it would be so unfair to say it was one technology that was like a magic bullet. That will not be true.
And I guess also, just from a field perspective, it was not just Moderna inventing it. It was a lot of academics. As you mentioned a lot of academics, and you mentioned Biotech. We mentioned CureVac.
Yeah.
I guess there's innovation coming from,
of course,
a bit everywhere.
Yes, of course. It happened in any industry. If you look at any industry over enough period of time, not a couple of quarters, but years or sometimes decades, you realize a lot of time helping an industry, there was a lot of players. I can remember the car industry when it started. There were hundreds of car companies just in the US, and same in Europe. And then now in the US, I think we have 3 car companies. I mean, maybe 4 now with Tesla. Exactly. And so you always see this phenomena, and we are taping this, I think, the day after Biotech announced buying CureVac.
Yeah.
So it's an example of exactly what happened in any industry, which is you start with a lot of smaller players, and as time goes by, some don't make it and disappear. Some get merged into other companies that are bigger. It's a natural process of capitalism, because to be able to, I mean, I just mentioned, we invest $200 to $400 million per year in just basic science. It's not clinical drug. It's basic science to keep learning about mRNA. If you invest $10 million per year, and even if you are twice as smart as we are, you're going to get less things done. It's just math. And if you compound it over 5 or 10 years, you're going to be way behind us, which is why in any industry, scale does matter. Look at it with hyperscalers in tech right now with AI. Look at it with car companies because you have all those fixed costs, the cost of designing the cars and so on. In any industry, scale matters, which is why I think given where Moderna is now, I think most of the future of mRNA for quite a while is set up. You have two big players, Biotech and Moderna. It will be almost impossible. I don't say that out of arrogance, just a kind of business history and practicality, which is the knowledge we've accumulated internally, the know-how in the last 14 years of a company. What we learned by having run literally thousands of animal experiments, by having 40 drugs in a clinic across 4 different technologies and so on. The amount of things we've learned,
Having those millions of people, I guess.
Yeah, and all those investments that we are making in research every year, if you were to start today and I think go see a VC and say, I want to start a new mRNA company, you must have a very, very good science or idea.
Even Neighbor will tell you.
Yeah, even about it. How are you going to catch up?
What's interesting on this as well is that, I mean, there was a few other mRNA companies, but actually not that many.
No, there was.
That raised the size. I mean, you were always like the 3 main ones. You were the 3 main ones.
Yeah, there was Efris, or still, I think they still exist, Efris in Germany as well.
Okay. Yeah.
There was one in Belgium, I forgot the name. There was a couple in the US which were like Arcturus. They tried not exactly mRNA like us, but self-replicating RNA. The couple companies in China, a couple companies in Australia, but they're all tiny. They're all, in my humble opinion, in today's world, totally subscale. So it's smart people working really hard, I'm sure. Some of them I don't know, but I'm sure working really hard and very passionate. But again, if you look at it from a rational business lens, a chance of scaling from that place, including just having access to capital. Even if you have the smartest people in your room, access to capital is going to be really hard.
As you said, raising 5 billion.
Yeah, I don't wish it on anybody.
You talked a bit on the other programs, but one thing that is also quite interesting was BioNTech. That BioNTech is quite focused on oncology.
Yes.
And was actually, before the pandemic, was more oncology players. You have always, I mean, you have quite a lot in vaccine. Even before the pandemic, you had quite a lot of vaccine. How, like, yeah, why did you have this different approach, or what the difference?
It's a very fair question. And I think to anybody who is curious about that question, I always advise them, because I get this question a lot from investors, as you can imagine, because both companies are publicly traded. And I always tell the same answers to investors. Say, spend 15 minutes downloading the S1, which is the legal document you file for an IPO of Moderna, and downloading the S1 of BioNTech, because both companies went public, as you know, before the pandemic. Read the S1s. You have in both cases the same management team, the same CEOs, and so on. And you will see that both companies, I think, you have to ask Ugur to make sure that I'm making the right comment about BioNTech, because I'm not there, obviously, and I'm not him, obviously. I think both companies have gone back to their pre-pandemic strategy because they did it for a reason. So our strategy since the day one discussion with Nubar was, because the field of biology has protein in every cells, in every disease state, there's 22,000 genes. We should be busy for a very, very long time, most probably longer than our lifetime, as a field to invent how to make mRNA medicine for people. I think BioNTech's strategy since early days of BioNTech, confirmed in their S1 document, is they want to be one of the most impactful oncology companies in the world. And I believed, I'm oversimplifying, again, ask Ugur, but I don't think I do it disrespect. I believe they don't really care about which technology they need to get there. Their obsession, which is very noble, and I'm rooting for them, obviously, is to have a big impact on cancer patients. And they believe that if to do the right thing, that the thing that they think is the right biology strategy for that cancer is to go with a small molecule and a CAR T and an mRNA, and then freeze after bispecific, they will develop that. And again, it's all to their credit. I think scientifically, I believe the same thing. If you look at our partnership with Merck, we partnered with PD-1 antibody drug. Our beliefs was that we don't need to have to own that technology to get it done. We can do it through partnership. So you see two very different strategies being put into music. Of course, there is some overlap. As you know, they did a partnership with Pfizer, which again saved so many lives at the beginning of the pandemic because they already had a partnership with flu. Because again, I believe they were not very interested in infectious disease before the pandemic. And they had partnered flu with Pfizer, which of course is one of the big four vaccine companies, to work on mRNA vaccine for flu together. And so I believe they're back to their strategy. And if you look at their earnings calls, and which of course we follow as we should, they are very, very oncology focused. And if you look at our strategy and how we're executing it, we are only mRNA focused.
Yeah.
If we're in a partnership, we go to people that have those assets. Because if we think there's a scientific rationale for combinations, it's the right thing for patients. We want to try it, of course.
I like that, yeah.
So
you don't feel like you're missing some opportunities that are, let's say, adjacent to mRNA or that are not using mRNA?
Look, I'm sure we have or we are or we will be. But, one thing you have to do as a company and as a CEO is to prioritize. Because there's just so many ideas that my scientists have and so many diseases still existing that you could go crazy doing 1,000 projects at the same time. But then nothing will move because you will not have enough resources and it will be such a complex enterprise to manage. It will just be so complicated that I don't think you'll be successful if you really care about impacting patients. So I think you have to choose what you do and what you don't do. Strategies is mostly, in my opinion, choosing what you don't do. Then you're left to what you do. And I think being then disciplined in execution to stick to your strategy. If things change, you can, of course, evolve your strategy, and you should if there's a change in context or hypothesis. Again, it's good science and good business. But I think that you have to prioritize, and that makes you sometimes miss opportunities, which might make you update or refresh your strategy.
Yeah, I like that. And actually, it's also what you said was Merck, was a PD-1. It's a good transition. I wanted to discuss a bit let's say, beyond mRNA vaccines directly, of like using mRNA as delivery of antibody or combination with antibody as delivery of bispecific. I saw the deal you did was Imatix, and we had Harpreet on the show actually.
Okay.
And I saw some was like in vivo CRISPR gene therapy or delivery, some cell therapy using mRNA. I think not Moderna, but some others like in vivo CAR T was mRNA. So how did you how do you look at it? Yeah, how do you look at it?
Sure. So I believe we're very practical people here, which is we want to make medicine. And so we try to invest in technology to make different applications of mRNA work safely in animals first and then in humans. So the vaccine, the cancer product, the rare disease, the lung inhaled product with cystic fibrosis that we have with Vertex. So we work on technologies that we think we can make work or that we sometimes stumble on and then make work.
Yeah.
Sometimes you have serendipity in science. Of course, you don't plan for everything. That would be arrogant to say we plan for everything. Sometimes we got lucky. And then we try to look at what's the Venn diagram with medical need at the moment. And try to say, to be able to get to this vision that we might end up a company in 20 years from now with 50 drugs on the market in many therapeutic areas, is what's the bridge to get there? And if we only dream about starting to get mRNA in the brain to cure Alzheimer, we might be dead by now. If this was a strategy on day one, because first, Alzheimer still has a lot of biology mechanism we don't understand as a scientific community. And I still don't know at this day how to get mRNA in the brain. And so one reason we went to vaccine early is we thought that our technology was ready for human testing. We were always very careful and thoughtful and worried about what are the things we don't know that could hurt people as we start our first clinical study. So we are very, very careful to check everything we could. And then to start a very low dose. And one of the things that attracted us to go into the clinic first in vaccine for infectious disease is because you don't need to make as much protein because then your immune system is going to see the protein made by the mRNA and make a lot of antibodies to protect you to future infection. The dose at which we started our first clinical trial was, I think, like 15 or 20 microgram dose, microgram, not milligram, not per kilo, but per human, not per kilo of human, but per human. So it was very tiny. So we most probably it's the least chance we have to hurt a person because, again, we are worried, given it's a new field. And then the other piece we liked about vaccine in terms of safety for the patient is because we are not going to make a human protein via mRNA in a healthy human, which is a phase 1. We have no risk of creating autoimmune disease. One thing I was so worried about is, let's say let's pick a very easy protein that people know. You go with insulin as your first one. You inject healthy volunteer in phase 1 at a low dose. There's something that happens in humans that you had not seen in monkeys or rats before. So it was unknowable this will happen. And the immune system of those human patients in phase 1 that are volunteers, because they're healthy, sees that protein made by the mRNA, sees this protein is weird for whatever reason we could not anticipate because it was unknowable. And that person's immune system makes an antibody to insulin and that person becomes diabetes. That would have been just, horrible outcome that we all have felt terribly guilty about to have hurt people. Again, we're in this business to try to help people. So last thing I want, or my team wants to do is to hurt anybody. So we go into the clinic always as a very, very serious decision that we make. It's not like a nonchalant decision we make in a hole, by ourselves. Say, let's go to the clinic. And so this is why, as we thought about all the things at the time that where the technology we thought was ready, we decided to prioritize vaccine first in the clinic versus rare disease, let's say, because we were really worried to create safety issue in healthy people that would have been terrible.
Okay. Okay, so basically what you're saying as well is that the beyond mRNA vaccine is there's still some that are relatively early and you will take it step by step.
If you look where we are today, we're in phase 3 study for cancer. We still have data next year, we said in 26. We're in phase 3 study for rare disease of the liver. We could have data also potentially in 26. As I mentioned, autoimmune disease, we are working in a lab in animals on autoimmune disease. It could get into a clinic into next year or two. So we're in a lung with inhaled mRNA, from the mouth with Vertex for cystic fibrosis patients that do not respond to Vertex drug today. Vertex has done amazing scientific work for patients to get drugs that work for cystic fibrotic patients, because before, of course, they had no hope. But the problem with their drug, because it doesn't work for every mutation of the CFTR gene of those kids. There's around 20-30% of the kids, based depending on countries and age.
The CRISPR therapeutics.
Yeah, they're also trying another way. But I think, again, the CRISPR angle, what I think over time, CRISPR is going to be an amazing technology. I wouldn't bet against human ingenuity. I think it has a lot of safety question marks, as it should, because as you go cut DNA, as we know today, cancer is always a disease of DNA. Look how paranoid I was and worried I was about getting into a human with mRNA that is destroyed after 48 hours in your body on a very dose of vaccine that doesn't touch your DNA compared to going and cutting your DNA forever.
Yeah. Good one. I think one last question before, so like quick fire.
Yeah.
You mentioned that you were already using machine learning in 2017.
Yeah.
And you did a big deal with OpenAI. I don't know how big the deal was, but at least it's a collaboration.
Yeah.
Can you talk a bit more on how do you use AI since 2017 for mRNA specifically and in drug development?
Yeah. So we need another podcast for your question because literally the answer is everywhere. So we use it in science, we invent new molecule, like the example I shared of a protein before. We use it in manufacturing to improve process yield, to help us do root cause investigation of deviation in the plant, to help us review and approve lots of products. We're using clinical trial like picking the dose for phase 3. We build the GPT. The clinical team build the GPT to help us to pick the dose. We use it in pharmacovigilance to analyze the complaints getting when people in clinical trial or in the real world have complaints about the product that might be or not be product related. We use it in commercial for marketing. We use it in HR. We use it in legal for contracting. We use it in finance. So that's what I'm saying. We can spend time in any one you want. The team has already written 2,000 GPTs within the company.
Okay.
And they are literally across everything.
That's a good one.
So we need another podcast one day.
Yeah, that's good. I will get back to you. Well, quick fire, just a few quick questions if you can. Like,
Yeah.
Yes, no, one sentence. What's on top of your mind at the moment?
How do we scale the company to go back to profitability in the next couple of years to redeliver on the mission?
What's the most difficult decision you had to make as CEO in the last 2 years?
Is to reduce the footprint of a company because the demand of products was coming down. So we had to
Down the hill.
climb down the mountain, yes.
One advice to young life science professionals who want to be in your shoes?
So if my shoes mean CEO, is learn about the science, if you're a scientist, learn about leading people because it's all about people, and learn about or be curious about financial markets, VC, because you're going to have to raise a lot of money.
5 billion.
Not necessarily 5 billion, but that was a big platform, Moderna.
What's your favorite biotech or science book?
I love to go back to old Louis Pasteur books. So I'm reading right now, old Louis Pasteur book is one of my hobby. I'm buying them in old bookstores for like $20. And usually I get originals and so on. And just to go back and see the amazing curiosity that he had. He revolutionized 4 scientific fields in his life. Just amazing.
I will check it out. One last one. One mistake you made in the past 12 months.
Oh boy, how long do you have? Because I mean, maybe one mistake, I need to think about one. I made people decision mistake. I made them all my career. Because go back to where we started with the question of fits. So yeah, I made people decision mistakes in the last year.
Maybe a last one more positive, but one new habit you adopted recently.
It's around using GPT. I deleted my Google search from my browser. So I put at the bottom of my iPhone the ChatGPT app. I mean, we have GPT Enterprise, of course, for security reason and our data. Is to start to always ask myself a question before I do something, like sending an email to somebody asking for an analysis or whatever. Is can I do it by myself with GPT and or using deep research?
I like that. Great. Thanks, Stéphane.
Thank you so much, Philippe. It was great to see you again.
Thanks. I'm impressed by how Stéphane and the Moderna team were able to deliver on the dream. I'm also impressed by Stéphane's humility and openness. If you also enjoyed this episode, please hit the like, follow, or review button. Any of these actions would help a lot the show to get to more people. If you want to support the show even more, you can make a donation via the PayPal link below. If you want to see similar videos, please check out our YouTube channel where we have many more. Also, I would be curious to hear what you think. So if you could leave a comment wherever you are, or shoot me an email at Philip@Flot.bio. All right, thanks for watching to the end and see you in the next episode.